In this video, we're going to be talking about animal virus life cycles, or the replication strategies of the different types of animal viruses. Before we get started, I want to ask the question, do animal viruses affect a eukaryotic host cell or a prokaryotic host cell? If you answer that they infect a eukaryotic host cell, you are correct. A key characteristic that's going to be essential in understanding these replication strategies for at least two of the viruses is the fact that a eukaryotic host has a nucleus while a prokaryotic host does not. So to begin this video, I want to draw a cell. So here is a basic picture of the cell. And this inner circle that I'm drawing right now is the nucleus. So within the nucleus, we're going to see the host genome. So we need to first look at the different types of animal viruses. We have double-stranded DNA. We have RNA, but we have to be more specific because we have both positive, or in other words, sense RNA. We have negative or antisense RNA. And we have a special type of virus that's also RNA that's called retroviruses. And they have a positive or sense RNA genome. So we're going to walk through each of these viruses and the replication strategies. So the first one we'll talk about is the double-stranded DNA. So the virus is going to enter in, and eventually it's going to uncoat. So it's going to let out its genome. The genome is then going to move from the cytoplasm to the nucleus. Once it is within the nucleus, it's going to use host cell machinery in order to convert itself into RNA. This process of DNA to RNA is called transcription. The mRNA is going to leave the nucleus and go to the cytoplasm. RNA is what is used to create proteins, and so the RNA is going to travel to the ribosomes, which are going to create the proteins in a process called translation. During translation, this is when you're going to get your protein synthesis occurring, and you're going to start building all those pieces. So we're going to get more capsids, we're going to get more spikes, and eventually all of that are going to come together through assembly to create a new virus. The virus is not going to want to stay inside its host cell. It eventually is going to leave, either through lysis or budding. So this is the structure and life cycle of a DNA virus. With the RNA virus, more specifically the plus RNA virus, positive RNA virus, or the sense RNA virus, it's going to come in here, it's going to uncoat, and the reason I prefer sense RNA is because, to me, it helps me memorize what happens. Because it is called sense RNA, it makes sense to the cell. So it looks like mRNA. So because it looks like mRNA, it can immediately go to the ribosome for translation. This will cause it to create more protein pieces, go into assembly, and eventually leave. So the positive RNA virus has a very quick life cycle, and it is the most simple of them. The next one is the negative RNA virus, or the antisense RNA virus. Because it is called antisense, it does not make sense to the cell. And so when it comes in, it actually has to convert itself into something that can be read by the cell. So it's converting itself from negative RNA to positive RNA. And the way it does this is that it actually brings with it its own packaged enzyme. So this is a viral enzyme, and that is what converts it from the negative RNA to the positive RNA. Once it becomes positive RNA, it's going to follow the other viruses, and it's going to go to the ribosome for translation. So I connected these lines together, so it goes to translation, makes more protein pieces, it's going to create itself, and then it's going to leave. This is the negative RNA virus life cycle. The last one is the retrovirus, which is very interesting. The reason being is because although it's going to have a positive or sense RNA, it actually does not go to the ribosome for translation. Rather, it actually turns itself into DNA. And it does this through a special enzyme. The enzyme is called reverse transcriptase. Breaking down this word, reverse means to go back. Transcript is short for transcription. Remember, transcription is DNA to RNA. And so ACE means it's an enzyme. So this enzyme does reverse transcription, or in other words, this enzyme converts things from RNA to DNA. Once it becomes DNA, it's going to follow the DNA pattern, and it's going to go into the nucleus. However, rather than being transcribed immediately, it's actually going to integrate itself into the host genome. This integration of the host genome has a special term. It's called provirus. So with bacteriophage, it's called prophage, but with animal viruses, it's called provirus. Eventually, it's going to be transcribed into mRNA. That mRNA is going to leave into the cytoplasm, and then it's going to go to the ribosomes for translation, assembly, and leaving. These are the viral life cycle strategies. If you need time to write it down, I recommend pausing the video. I recommend also going through all three videos to get an understanding of the differences and similarities between animal viruses, bacteriophage, and the general strategies that are seen with both.